Hello, ninth graders. I hope you're doing well and that you had a great summer break. This is Miss Fatin, and I'm excited to be your biology teacher again this year. I hope we have a fantastic year together, enjoying our biology classes. Today, we're starting with a short lecture to revisit the digestion topic we covered last year. This will also be our focus for the first 4 to 5 activities in the book this year. After that, I'll go over the review worksheet, especially the action verbs we've covered. So what about digestion? We know that it is the breaking down of complex food molecules into simple food molecules known as nutrients. What are examples of complex food molecules? We can mention the proteins or the polysaccharides. What about the nutrients, which are the simple molecules that can be used by the cells? The amino acids, and the monosaccharides. So through digestion, proteins are turned into amino acids and polysaccharides are turned into monosaccharides. Digestion happens in our digestive system that consists of 2 big parts: the digestive tubes with all of its organs, and the digestive glands, which are the accessories that are well needed by the digestive system to do digestion. Digestion includes both mechanical digestion and chemical digestion. When we say chemical digestion, we're talking about enzymes like the salivary amylase, okay? And enzymes are specific. Do you still remember the 4 properties of enzymes that we talked about? Enzymes are specific to substrate, to temperature, to pH, and to their duration of digestion. Let's talk a bit in deep about all of this. So digestion is turning complex food molecules into nutrients that can be used by the body cells. Look how complex the substance is in here. Through digestion, it is cut into simpler substances that can be used by the body cells. Okay? Examples of a complex food molecule is the starch molecule found in bread, found in rice, found in pasta. Okay? Starch is an example of polysaccharide. It is a complex kind of sugar. Okay? After digestion, it becomes small or simple molecules of sugar. Okay, look at this. Starch found in bread is turned into simple sugar molecules under the effect of amylase, which is the enzyme. Okay, this is chemical digestion in here. Protein found in chicken and meat and eggs is turned into amino acids under the effect of an enzyme, protease. Fats or lipids are turned into fatty acids and glycerol under the action of lipase. Lipase, protease, amylase are examples of enzymes, okay? And simple sugar molecules, amino acids, fatty acids, and glycerols are the nutrients that the cells use to perform different activities, to make energy, and so on. Our digestive system, where digestion happens, includes, as I mentioned before, 2 big parts: the digestive tube and the digestive glands. The digestive tube consists of the mouth, esophagus, stomach, small intestine, and large intestine or intestine. Take a look on this. Okay, here we have the mouth. So the digestive tube organs are the organs where food passes. Food goes through the mouth, then through the esophagus to reach the stomach. Then it passes to the small intestine and ends in the large intestine. Food passes through the digestive tube, but it does not do that through the digestive glands. The glands are the accessories connected to the tubes, and they are responsible of making digestive juices that contain enzymes to do digestion. Okay, the salivary glands are connected to our mouth to secrete saliva. This saliva is the juice that contains salivary amylase, which is the enzyme needed for digestion. Okay, so salivary glands in the mouth. We'll go over them more in details in a few. Gastric glands, gastric yani connected to our stomach. Okay, the stomach is connected to the gastric glands that make the gastric juice containing enzymes. And then we have the pancreas, the liver, and the intestinal glands. They are all connected to or linked to the small intestine. They secrete their juices there. Okay, the juices secreted by those glands are responsible of the, not the mechanical, the chemical digestion. They are responsible of the chemical digestion in the body. Okay, what about the mechanical, you'll say? So chemical, it is done by the enzymes. Mechanical, like chewing, like mixing food in the stomach. Okay, mechanical, we are just turning the food into smaller pieces. But for example, the mechanical digestion of starch results in smaller pieces of starch, but it does not turn starch into monosaccharide. Okay, chemical digestion is done by the juices secreted by the glands. Mechanical digestion facilitates chemical digestion. It makes it faster. Okay, chemical again is done by the enzymes. Okay, the enzymes found in those juices. Saliva is a digestive juice secreted by the salivary glands in the mouth. The gastric juice, it contains pepsin, if you still remember. The gastric juice is released by the gastric glands when in the stomach. Small intestine is connected to the pancreas. Liver will intestinal glands. Pancreas releases the pancreatic juice. The liver تعمل البايل. The intestinal glands بيعمل intestinal juice. Okay? These three juices have enzymes in them to do the chemical digestion. Okay, it is important to note that chemical digestion happens in the mouth, stomach, and the small intestine. Okay, this is a general overview of the digestion topic we covered last year. Let's move now to the questions of the worksheet. Question number 1, indicate the true statements and correct the false ones. First, we have to tell whether the statement is true or false. And then if it is false, we must correct it. Okay? Look well at statements A and C. They talk about tests, food tests. We studied 4 different food tests. Ilna, we have the biuret test, the iodine test, Fehling test, and the translucent spot test. Each one of these tests has its own function. يعني البيوريت test helps us to identify the presence of proteins and or polypeptides. الأيودين test هذا البيوريت. الأيودين test to detect the presence of starch. Fehling test for the reducing sugar. قلنا كل الsugar that we studied هن reducing sugar إلا sucrose. Translucent spot test to detect the presence of lipids. So it depends on what we want. And if the test is positive, this tells us that there is this kind of food. Oh, there is not. There is this kind of food is not present. Okay. The second statement talks about mechanical digestion. We talked قبل شوية عن two types of digestion, mechanical and chemical digestions. وكمان the last statement is saying low temperatures lead to the inactivation of enzymes. I'm not going to solve the question now. I'm just trying to clarify the statements, okay? So statement A, the biuret test detects the presence of proteins and or polysaccharides. Proteins is something and polysaccharides, يعني complex sugar is something else, okay? second statement, mechanical digestion breaks down food into small pieces. A third statement, the iodine test gives a brick red precipitate. Do you think that? Which test بيعطيني brick red precipitate in the presence of a reducing sugar? So there is one of these tests is used to detect the presence of a reducing sugar. Yes, it does give a brick red precipitate in the presence of this kind of food, but it's not the iodine test. Actually, the iodine test turns from an orange color, brown color, into a dark blue color in the presence of something else. Okay, as I told you, I'm not mentioning the answers in here. Low temperatures lead to the inactivation of enzymes. As we said, enzymes are specific to what? To the substrate. So مشكل enzyme works on all substrates. لا. Enzymes are specific to pH, and some of them work in a neutral medium, others work in an acidic medium. Okay, they are specific to temperature. So what do you think the effect of low temperatures on enzymes? This is question number 1. Akid will find the answers in the solution, and we're going to solve this anyway in class. Question number 2, the following experiment aims, so here we have the purpose of the experiment. We are studying different enzymatic properties. Salivary amylase is used as an example of enzyme, akid. It is found in the saliva. Wheat bread is an example of food rich in starch. Okay? Question A, part A, indicate. Whenever you see indicate يعني just tell, يعني بس give the answer, do not justify. Tell me, what is the variable factor? شو ال different factor بين A و B? Look at A و B. هون في bread, هون في bread. هون 37 temperature, هون 37. pH نفس الشيء. We look at the conditions. What's different in here? Here we have the salivary amylase, هون عنا البepsin. So what do we call this? How can we mention that this is the variable factor? بعدين بpart B في عندي interpret. Interpret يعني analyze the experiment. Okay, so as I said, indicate, no justification. Interpret, you analyze the experiment, بعدين this indicates that. شو يعني analyze? Analyze يعني I have to say what we did بالexperiment وشو results, and we should not forget on any of the conditions. يعني I have to say إنه حطينا البريد with the salivary amylase at 37 degrees على pH for 60 minutes وطلع عندي هالقد bread. Okay? They're asking us to interpret both experiments A و B. برجع بقول بالB عملنا 1 2 3 نفس الشيء, and we connect these two using شو? while. Okay? Why? هلا هيك بكون أنا I did the analysis part. Am I done? لا. I have to add this indicates that. This indicates شو? How to put the indication. As I always tell you, you go to a scratch paper. بتحطوا شو الفاريبل هون. الفاريبل, I'm going to answer this anyway. الفاريبل هون الانزيم. يعني بالA we use salivary amylase, بالB we use pepsin. How did this affect the results? With pepsin, the bread remained 100%. ماشي صار للبريد. Digestion did not happen. بقي البريد 100%. بس بقي البريد اختفى. يعني نحن when we changed this, results تغيروا. This is what I have to mention بالindication. بدي أحط variable factor with the result. يعني this change in enzymes caused digestion to happen أو not to happen in tubes A و B. Okay? بعدين conclude the enzymatic property. One of the easiest questions هي موضوع enzymatic property. هون وهون. There are 4 enzymatic properties. Listen well. 1, each enzyme is specific to, each enzyme acts at a specific substrate. 2, each enzyme acts at a specific temperature. 3, each enzyme acts at a specific pH. Okay, well, for each enzyme has its time to digest a substrate. تمام? So the answer B و C will be one of these 4 answers. هون نحن we changed the enzyme. يعني أكيد الانزيماتيك property بدها تحكي عن specificity between enzymes and substrates. Okay. وهون بالC, let's see what we changed in C. طلعوا بالC we use the right enzyme. يعني حطينا bread مع salivary amylase. برافو, it's the right enzyme لأنه salivary amylase هو اللي بيعمل digestion للستارش بالماوث. Temperature, it's the body temperature 37 degrees. بس شو عملنا pH 2 صار acidic. okay. Positive iodine test. شو يعني positive iodine test؟ يعني there is starch. Okay? Starch remained. It did not disappear. البريد بقي مثل ما هو. يعني salivary amylase ما اشتغل. Okay? We have to specify whether digestion happened. أول شيء بقول my answer. I indicate my answer. إنه yes, digestion happened in tube C. No, digestion did not happen in tube C. Okay? Look carefully at the table وأنت قول digestion happened ولا مش happened. Did bread disappear ولا بقي البريد ما انعمل له digestion. This is what I have to say first. Specify يعني I have to justify. بقول له since. Okay? وأنا أكيد دايماً I prefer when you justify, you analyze the experiment. يعني since when we placed bread with salivary amylase at temperature 37, pH 2, we got a positive iodine test showing that starch remained, the bread remained. ما اختفى الستارش. Digestion did not happen. شو الانزيماتيك property؟ أكيد something related to the pH. تمام؟ Again, you will see we will solve this in detail in class, or you can check the solution. Question number 3, which is the last question behind the worksheet, we have a graph, actually 2 graphs, okay? The first one showing the quantity of nutrients in the small intestine. How many times is this quantity changing? It is just decreasing. تمام؟ وهون the second graph, it is showing the quantity of nutrients بس وين بالblood leaving the small intestine. اوكي عم بيقول لي analyze the results. دايما نحن بنحط as حد اللي تحت، حد الفاريبل اللي تحت. اوكي we have a dependent variable. Dependent variable هو يلي فوق. يعني أنا بقول the quantity of nutrients in the small intestine شو عم تعمل؟ decreases. It is analysis. يعني you have to mention the numbers. You have to mention the units. Okay? What are the numbers I need to mention؟ بطلع بالجراف بلاقيه هو بس عم increase. بس عم ينزل. So قول the quantity of nutrients in the small intestine decreases from 90, and أكيد I have to mention the unit, 90 AU at T1, okay, to 2 AU at T4, okay? Or I can say it decreases from 90 to 2 as time passes. شفتوا بحط الأس حتى اللي تحت. As time passes من T1 to T4. Did I finish؟ لا لأنه I have another variable that is changing. يلي هو the quantity of nutrients in the blood leaving the small intestine. كمان مرة عم بعمل analysis ل two things together بحط بيناتهم while. Okay? While the quantity of nutrients بالblood leaving the small intestine شو عم تعمل؟ بس عم increase. Okay? هون بس decrease هون increase. So باخد أول نقطة وآخر نقطة. It is increasing from 10 to قد إيه؟ 98 as time passes من T1 ل T4. Okay? بquestion number 2, so أول واحد هو analyze. Second one بدي احول الجراف لتيبل تطلع how many points انا I have. عندك اكيد حيكون التيبل عنده 3 rows. اوكي the first row اللي تحت بتصير فوق. يعني اول row رح تكون للتايم. second row رح تكون اذا بدكم للquantity of nutrients. مرة بالsmall intestine مرة بالblood leaving بس the small intestine. اوكي so 3 rows. شو هن البوينتس اللي انا بدي احطهم؟ بالسمول intestine عندي هيدي البوينت 1 2 3 4. بالبلاد leaving the small intestine كمان عندي 4 points اللي هي هاي وهاي وهاي وهاي. Okay. Derive a conclusion. Can you see إنه the quantity of nutrients بالsmall intestine هن النيوترينتس وينهم بالsmall intestine. النيوترينتس بالsmall intestine عم بديكريز عم بقلوا. طيب وين عم بيروحوا؟ عم نشوف إنه the quantity of nutrients عم تكبر بالبلد. يعني نحن فينا نقول إنه هدول النيوترينتس عم بيروحوا من الsmall intestine على البلد. What do we call this process? It is the intestinal absorption. تمام؟ يعني أكيد after digestion finishes لازم هدول النيوترينتس to share them with the body cells. مين اللي بيعمل هيدا الشيرنج؟ هو البلد. Okay? See you inshallah in class. We're going to solve this together, and I'm going to upload the solution anyway on the application. And if you still have any questions about the worksheet or the lesson topic, feel free to ask. See you.